we can discuss later.
I'm glad. Well, no, it's practice.
I think they want to start. We can talk about it over lunch.
Mr. Broccoli. All right, we have the next talk, Siobhan from UCLA, talking about cell shapes.
Hello. So, not animals is the first part. We're going to talk a little bit about plants. And I sort of want to, I'm going to frame a question for you, but I'm also going to frame who I am, which I think is useful. I am a developmental biologist, trained developmental biologist in plants. But I am sort of a microbiologist now, and you'll sort of see why that is true. I'm interested in morphogenesis in plants, the generation of shape and pattern, and I consider that to be a fundamentally physical problem, which you'll also see why. But in the course of that work, what we have to do is, if we want to describe how shapes are generated, obviously we have to also be able to describe shapes, and also the diversity of shapes. So I am not an evolutionary biologist, so this is something we're just getting into, so please be forgiving to the expert evolutionary biologists. So this is a plant. You see them all the time. You've got one of the interesting things that's different about plants and animals, to me, is that you get these really striking patterns that actually get generated throughout the lifetime of the plant. So when you have a seed, it's not as if you have every single leaf or every flower that the plant will ever have in that tiny little shrunken seed. Instead, they're actually going to form these organs as they grow throughout their lifespan. So you have continual development, continual pattern formation, continual changes in shape that occur, and generation of new shapes. So this is something that I've worked on before, which is the arrangement of leaves around a stem. It's called phyllotaxis. And we get really quite precise mathematical arrangements of organs around the stem. And we've done some work looking at how that's patterned actually up at the top of the plant where the new organs are being formed. But there's also diversity in not just the patterns of where leaves are, but also, for example, leaf shape. And we saw a short example of this. Well, it was flashed up there yesterday that persistent homology, for example, is something that's been used to look at leaf shape and is ongoingly used to look at leaf shape. But leaves themselves also have really interesting margins, and that is a shape that you might explore. But there's also these patterns of where the veins go in the leaf, and that is patterned as well. And so all of these levels of shapes and patterns that you could choose to describe and actually ask questions about how they might be linked to each other as well. And I'll sort of explain why we think they might be physically linked. So this is actually an image of a bean leaf surface. So if you take any plant, this is not a bean, but you could take this plant, you could look at the surface of its leaf, and what you would see are cells that look a little bit like this. They have sort of an individual cell looks a bit like a puzzle piece stuck together. It's basically a jigsaw puzzle, but it's very frustrating because there is no sort of pattern that you can match. These cells here, stomata, which you may be familiar with, they're the reason they'll open and close and allow the plant to exchange gases so it can photosynthesize and basically make all of the carbon into solid state so that you can eat it. It's very useful. But I'm going to kind of focus on this pattern today. So not necessarily the tiling of the surface, although that's an interesting question, but just how do you get sort of undulatory margins? And this is, potentially pertinent for the field because this is an example of one of our model species, the Arabidopsis thalamana. It's a weed, but we can do a lot with it. We can do a lot of genetics and developmental biology with it. And you can see here that it has these undulatory cell margins and they're fit nicely together. In our other model species for this same system, corn, maize, you have a more elongated cell shape, but you also have undulatory margins. So this is a scanning electron micrograph, as is this, just slightly different. I don't have scales on there at the moment. I'm going to have scales a little bit later when it becomes important. So the two model species that people use in plants to study the morphogenesis of this cell shape in the epidermis both have undulatory margins. And this sort of, I think it's a really cool question, and so actually I'm going to show you how we're working on how these undulations might arise morphogenetically. But we also just wanted to ask whether or not this was actually the way that most cells look like on an epidermis. If you literally grabbed a leaf from anywhere, would the surface look like one of these?
Can I ask a question? Each cell is at the boundaries when it connects to another cell is basically going into the board, so it's becoming thinner in the third dimension, or?
Yes. So the question is whether or not sort of in this third dimension, are they sort of pinching down where they're connecting to each other? And the answer to that question is yes.
And it's all genetic, always going to be at the places where they come together, it pins down?
Yes, and that's because they're physically connected to each other. And there is I believe, we have to measure this, differential growth at differential faces of the shape. And so there is constraint on the boundaries in that third dimension, which does not exist here on this free face, and that will lead you to sort of bulge out the plane, which may be a mechanism by which some of this
You don't need differential growth, you just pressurize which cells are, and you have more
Yes.
cell wall at the boundary, that's enough.
And remember, this will become important later, this is a free face also, so all these top walls that you're looking at, they're free, that surface of the cell, whereas underneath, which you can't see, and all the sides are connected actually to their neighbors. So that also would allow you to bulge. There are some differences actually in material. So these surfaces tend to be thicker, and they may have material composition differences as well. But sorry, just along that line, if you do depressurize these, so you have to think about a plant cell unlike an animal cell where its structure comes sort of from its cytoskeleton inside of the animal cell and attaching to sort of other things or surfaces, like a person inside a bag. That's how an animal cell gets its shape, and it's dynamic. In plant cells, the shape change that you see is plastic, it's non-reversible, and it's due to changing actually in the shape of the cell wall. It's like a box within which the cell contents live, and they can become highly pressurized because they're pushing up against this sort of surface within which they're enclosed. Does that make sense? So the way you generate shape is fundamentally different. So morphogenesis becomes a different problem. Is that helpful? Okay. So this kind of gets to this question. I will point out that for the most part, the shapes you saw on the last slide are generated from a field of roughly isometric sort of cubes, so I should show the squares here. And then they're going to start expanding. The box is expanding, the box is changing, and so while we're going to be looking at individual cells, you remember that as the shape develops, they're not developing alone. They have to have some coordination with their neighbors. And so what a lot of what my lab works on is how the material of that box changes in order to allow for a shape change. So if you actually had a cardboard box, and you had a balloon inside it that you'd blown up, that's providing some force for shape change. If you wanted that shape to actually change, you might need to sort of soften the material of the box in one area so that it could distend, and that would give you, say, a bulge. Now, the biochemistry and mechanisms by which the material changes we're investigating, but also what material properties lead to change is something we're also looking at. I'm not going to talk about that today because I'm going to focus on our analysis of the shapes themselves, but it becomes relevant when we ask questions later about how the shapes are generated. Yep.
I'm just curious why the cells do this. Like, why do they create an undulated morphogenesis? Do they need more surface area?
I certainly hope that I might be able to give you a small, unsatisfactory answer to that question. Yeah, because so our model species have undulation, and people make all kinds of claims in the literature about how important undulation is. I should study this. You should give me money. You should care about this because it helps for surface interaction, either sort of chemical signals moving across or even physical interaction, right? So this would be a more mechanically stable interface than this. Okay, so there's lots of arguments out there in literature. I hope I'm going to give you some insight into what we think might be happening. But in order to do that, what we really had to do was look beyond just 2 species. So we know of 480,000 species of plants. Those are the only ones we know about, and that's probably an underestimation of the number that exist. So I'm going to show you a sampling of 278 species, which we purposely sort of took from around the phylogeny, but it's still a really low sampling of what's out there. But we wanted to see what kind of diversity in the cell shape existed. So what is the actual morphospace for leaf epidermal cells if you kind of sample across the plant phylogeny? As best as you can without like thousands of people to do the work for you and access to bazillions of samples all over the world. What, if any, function might be implied? That gets to your question. And then I hope we have some time to sort of look at what power for understanding the morphogenesis might actually lie in some of the pattern variation. And then I hope I can tell you about an example of sort of 4-dimensional shapes that we're interested in looking at. So we looked at 270 plant species, as I mentioned, to see what their epidermal cell shapes look like. And this is work that I did in collaboration with my good friend Madeline Morlett, who is an evolutionary biologist in plants, and I'm actually an undergrad in her lab who's here, Grace. She's now going to grad school here at Harvard, did a lot of this work as well. So it's a collaborative effort. So here's some examples of the morphospace, not quantitatively, but just qualitatively. These are examples of the kinds of cell shapes that we saw, and these have been scaled for area. But there's obviously a diversity in undulation. So not all margins are undulatory. But we needed to quantify that, and so I'll just point out also, you can get all of the images and like the cell coordinate outlines and things on Dryad. If you want that link, you can email me later. We've put it all up there for free, so that people can use it and do fun things with it.
Awesome. I know she's going to use a square root again, but they don't want to give it for free.
Don't tweet.
Tweet away. Like, I don't mind people using this data. We have other questions, I think, that we're going to, so people want to use it if they can. So then, for us, we have this question, okay, well, how do you quantify shape? Because plant biologists have been really good at qualifying shape. It's undulatory. It's more or less undulatory. And we wanted to come up with ways to talk about that. So I'm going to show you some really low-level morphometrics and explain how we're trying to get a bit better, but also hopefully you have some suggestions. And I'm going to start by talking about this as a 2D problem, but it is a 3-dimensional problem as well, which I think is fun. Okay, so the traditional morphometric descriptors that we ended up using, most of the traditional morphometric descriptors, but these were the 2 most informative. So aspect ratio, basically the width and the length of the cell, and so the cells are always, to the best of their ability, registered based on their longest axis. So the outlines, because they don't really have a real registration in the leaf, but they're aligned along their longest axis. So we look at aspect ratio, and we also look at solidity, which is a convex Hall measure. So essentially what that means is if you were to sort of stretch an elastic band around the perimeter of the cell, and you looked at the area encased by the elastic band versus the cell itself, the closer that gets to 1, the less undulatory your own margin might be. This is not a perfect measure. So as you will see, we're trying to develop new things. So,
it is not the convex hull.
Yeah, some of those were not convex.
sorry. Yeah, these are not generated by an actual mathematician. These are just things that I sort of drew to illustrate it. So I apologize if they're imperfect. These representations are not real. The shapes are, but not this. That was, yeah. The math is real. So we used a real morphometric program to give us the solidity value. Later you can tell me exactly what's wrong with those. And then we'll fix them.
This is artist's rendering.
Yeah, like with a lot of biological license, which means like 98% wrong. Okay. So the reason that we sort of looked at solidity and aspect ratio is because when we put all of these morphometric parameters for an entire data set into a principal component analysis, we ended up with a morphospace sort of described by these vectors. And I'm showing you the first two principal components here, which explain, roughly 75% of the variation in shape. So this morphospace is the major contributor to what we're looking at in terms of difference. I'm just showing you two large groups of plants, so you can sort of see how this space sort of differentiates for them. And here, solidity and aspect ratios seem to very nicely separate the data in sort of perpendicular directions. And circularity is it's influenced by both the solidity and the aspect ratio, because this is how close you are to a circle, which will change with your undulation, but also your anisotropy. Does that make sense?
I'm trying to understand what you mean by solidity.
So the solidity here is the area of the convex hull divided by the area of the cell.
The other way.
The other way, area of
Okay,
the cell
sorry,
divided
okay.
by the convex hull. Yeah. So that circularity in some ways is influenced by both of these parameters. So we decided it wasn't very specific. These were slightly more specific. So
Can I ask if you are also going to tell us about, maybe I'll just wait, but ask a question about long-range order. Are you going to look at individual cells? You're also going to look at how correlations of orientation at one location compared to other change as a function of distance between points?
So I guess the question, which I think is an interesting one, is if you took a leaf, say the leaf had this particular shape, and you were to examine, cell
Yeah.
shapes here versus here versus here. I mean, this is not
Yeah.
too different from some of the skull images that actually had up. So we have not done this yet. So our sampling was almost always done. You tend to have a mid-vein, not always, but our sampling was done sort of in a central area of the leaf. We tried to be relatively consistent between species. We have not sampled across the entire leaf yet. But that would be an interesting question, especially when you ask things about how do you tile a surface or how do you grow a surface when you're trying to relate between the scales of the cells and the scales of the leaf itself.
And in time as well? In
So-
principle?
It would be beautiful to be able to look at the development of the shapes in time. With respect to that, we're a little bit limited in our methods right now. So, and also things being sort of more high throughput. So if you want to look in time, it's very handy to have confocal images where you have a fluorescent cell perimeter marker and you can look at that over time. Gives you nice contrast, which makes automatic segmentations easier. We can do that very well in only a few species where we can make transgenic plants. We can use fluorescent dyes, but then we're limited by how much time and money we have to pay for the confocal microscope. So we're going to ask these types of questions, but we're going to zoom very narrowly onto a small phylogenetic range for this functional question. So this broad sampling was very much done to just say, in general, what do they look like? And then we want to start addressing some of these more specific questions. Okay. So if you look at the distribution of these two, you see something sort of like this. So solidity is very much long-tailed. And where the star is kind of where our model species sit. So in general, these are quartiles. These are sort of 25 cells around the median quartile. So you can get an idea of what things look like in each quartile. And so very undulatory things like our model species actually are quite rare in the population. So saying that this is a super cool shape and we should study it because maybe it's really important is perhaps a mildly weak claim. But it also means that you have a continuous kind of distribution of the trait. So you're not looking at things are undulatory or they're not. There's different degrees of undulation that you can see exhibited, which is interesting if you think about how that might be patterned. This is aspect ratio, and you can see there's something similar. Different plants fall within this, but it is slightly more normal-ish type distribution than you get with undulation as proxied by solidity.
Are there ecological correlates?
So all of our samples were taken from 2 botanical gardens. So one in Cambridge, United Kingdom, and one in Amherst, Massachusetts. And also a glass house, I think, right, Grace? You did some glass house. Or was it all outside?
Oh, no, most, yeah.
Most of them were inside, yeah. So they're not grown in the same habitats as they would have been. And I think also, again, this was such a broad sampling that it's very hard to say whether we have ecological correlations. But this is exactly, again, just like this question over here, something that we want to narrow down on a particular branch of the tree that has a well-resolved phylogeny, some related species that have diverse ecological adaptations to see whether or not there's any correlation there are some old ideas in the literature that, that the levels of light you have might influence this trait, and we want to be able to test those too, but in this huge data set, that's not possible. Okay.
And the star is just a star shape where you.
Yes.
that's what-
I just want to point out that that's where our model species lie in these distributions. So it's not because they have a star shape. You can get star
Because
shapes
it could
in
be
plant
there.
cells. Yeah, it might
In
fall
terms
in.
of.
In terms of solidity, it might fall.
Yeah, the aspect ratio would be higher.
So can I
Sure.
Yeah.
So in the solidity measurement on the previous slide, the shape of that violin plot, if I'm interpreting the measurement correctly, it means that the variance is greater the more solid they are? Is that right? I would have expected the reverse.
Yes, so, no. So that was just showing how many species lie within a particular solidity range.
So is there a pattern that relates variance to solidity?
So I have the correlation plots in general for those. And there's, the more undulatory you are, the more variable you are. That is true. But it's surprisingly not a very strong correlation. So there's a couple species where there is an incredible correlation. And we've looked a little bit at within species versus between species variations. And maybe I can tell you about that after the question. So one of the conclusions here is that the model species for undulation are sort of rare and not really representative. So if we want to talk about how those undulations are generated, we should probably consider the fact that they are not that common, that there is this continuum of undulation that you might see. So we can ask any mechanism that we might come up with to be able to explain actually the entire morphospace. And that's a way to sort of test how good your morphogenetic models are. How do these traits look like when they're actually tested within a phylogenetic context? So this, the idea behind this was, well, I'll get to this in a second, but if something was really functionally important, you might expect that once you had it, you would hang on to it as a trait. So this is kind of a complex diagram. I'm going to walk you through it with respect to undulation. You have some groups of plants, so the most basal ones that we have are ferns. We then have gymnosperms, so these are things like pine trees and things like this. There's some early diverging angiosperms. Then we have now, those are flowering plants, monocots, so these are a lot of grasses and things like this. And then these are flowering plants, but you don't really see their flowers. Grasses aren't very beautiful to you. These are, over here in green, are what we call the eudicots. So these are the ones that have the really gorgeous flowers that we see all the time, that display much more complex pollinations and go through animals and things like this. But their leaves, this was what we were interested in, leaf shape. So what we can see, and this is basically on a phylogeny that exists based on genetic relationships and sequence relationships, we're mapping these morphometric traits. And we're asking whether or not there's a strong phylogenetic signal in any group. So for example, in ferns, it's kind of hard to see with these shapes, but fern cells tend to be highly undulated. So they tend to be in that lower quartile range of solidity. So they're very, very undulatory. Yeah?
Are your terminal nodes here families?
So the terminal nodes here are the species that we sampled. So what you're looking at is not a phylogeny of all the species, but our species extracted from the existing phylogeny. So you can sort of see the broad sort of sampling we had, but basically you tend to see phylogenetic signal in things that are really closely related. but not beyond that. So you're getting, if it's broccoli, you're seeing your relationships up at the tops of the broccoli, but you're not seeing a lot of conservation of relationships deeper into the broccoli. So the details of this perhaps are less interesting at the moment, but the ferns would be an example of where deeper layer broccoli correlation exists. So most of this branch has really undulatory margins and then a significant signal for undulation as proxied by solidity. But that's really the only place we see that. Interestingly, monocots, which is where maize lies, they actually have a high phylogenetic signal for being lowly undulate or for having a high solidity value. So they shouldn't, maize is a weirdo in that it's undulating at all. Most of those monocots sort of here tend not to undulate at all. So it's a question about what species you choose to look at when you're trying to model something.
If you go back and if you look at, I guess, like the old versions of corn or maize, like I forget exactly what it's called, but do they still undulate or are they less undulated?
The old versions of
Teosinte?
Yeah.
Oh, teosinte.
Teosinte, yeah.
Someone asked me that the other day and we haven't looked.
Okay.
So this is before domestication, which corn is domesticated and has been very much changed architecturally and developmentally by human intervention. Teosinte is the common ancestor that still sort of existed and probably where modern maize came from, but we haven't looked. I think that's an interesting question. The most closely related things to maize do not show undulation, so is it something that came up in the domestication process? I can answer that again. What's interesting is that the eudicots, this really diverse area of flowering plants where you have just an insane amount of species. The flowering plants dominate 95% of our known species, and a lot of those are eudicots. And this is one of Darwin's abominable mysteries, the rapid, incredibly rapid evolution of these species. A lot of that is thought to be due to their flowers, but we don't know. But it sort of means that all of these guys in green, really, these are the things where if you picked up a leaf, you would have literally no idea what to expect if you were to look at the leaf epidermal cells, because there is no strong phylogenetic signal. And so there's a lot of caveats for this statement, and I think we need to test this statement, but it kind of implies that, if there was a single optimal function provided by this high undulation, you might have expected that it would have been retained during evolution. Like in the broccoli, it might have popped up, and you would have then seen that kept. And maybe that's happening in the ferns. But we have a very sparse sampling for this, so we'd like to do sort of a more in-depth analysis looking at a particular clade. So what about function then? So this idea of a single optimal function I think is interesting. If you remember, plants grow and develop their shapes throughout their lives. So it's not like they're just constrained to this narrow window of embryogenesis and embryo development. And so perhaps they actually have more space to innovate and redeploy modules and change things and optimize functions throughout their life, not just at that one window. Yeah.
Is there any correlation between the modulus of the walls itself and the undularity? Because I don't know, I just have a feeling that the ferns might be leaves might be stiffer or I don't know.
So the question is whether or not there's a correlation between, say, the elastic modulus of a cell wall and its undulatory nature. And this, you could think of this as a buckling question, maybe. So, or just the deformation of an elastic rod, right? And depending on the modulus, you would get a different load, different wavelength, if I'm not mistaken. We don't have data for most of these with respect to what the elastic modulus of that wall might be. But that is something that my lab does. So it is something that we're interested in doing. And I'm going to sort of look at that in terms of maize in a second. Yes?
For the graph that you showed before, for the microscopes in the same time, even you see very different examples.
Yeah, so even within sort of the same family level, you can see some very wildly different examples. When we had sister species, so really closely related species, they tended to be similar. But after that step, it completely degenerated. The relationship didn't exist. exist anymore. So, I mean, a couple things sort of, if you imagine that there's a single mechanism by which this morphogenetically is generated, it means that that mechanism can actually be tweaked quite easily to give you this array of phenotypes, and that it is maybe perhaps not terribly constrained. But I think we have to test that a lot and more in depth. So there's been a recent proposal where they used a little bit of phylogenetic context, and that's that possibly, undulation exists to minimize geometric stress in large isometric cells. So here's an example of the finite element simulation that they did. Note that the material properties of these objects are uniform and identical between the different objects, and all they're changing is the geometry. And then they're looking at the maximal stress distribution within a finite element simulation. So here, as this cell just grows larger, so this is isometric expansion, you're seeing it's accumulating a large degree of stress due to that shape. Whereas if you undulate, you can sort of minimize some of that stress. Yes?
Has any of the plant breeding communities studied kind of undulations and heritability of it or variation of it across, different lines?
So the question is whether anybody who's done plant breeding has looked at this trait specifically, and I would say no, because they're usually maximizing for ergonomic traits. And so it's possible that this morphogenic trait segregates with some sort of agronomic trait, like maybe they are better at retaining water, and that would have affected photosynthesis. And if they were breeding for photosynthesis capacity, then they might have picked up a mutation that gave more or less shape. But we don't know that. There are some populations we can look into.
It seems to me that to answer this kind of question, you also need to know something about the constraints on area versus volume. So I'm puzzled. Well, I've not read this paper, so I don't know. But that surely should be a tuning parameter that these people.
So I would say that would have been really nice to have seen in this paper. It wasn't necessarily. I mean, for me also, as someone who thinks about sort of the material of the box, I don't think that it's, it doesn't really make sense to have the same material. But I think it's an interesting proposal. And sort of what they looked at some species, but sort of very closely around that model species, Arabidopsis. And they saw this type of relationship. So they made a metric called lobiness, which is essentially solidity. So just perimeters instead of areas that they're comparing. But it's pretty much the same. It's a potentially more sensitive to error, I would say. So basically what you're looking at here is these are sort of cell areas, so ranked by cell area, and the number was sort of how loby they were in another graph. So the most lobiest would have been this one, and then this one, and then this one. So the things that were the sort of 4 things that were most loby happened to be in this higher end of cell size. And the things that were less loby happened to have smaller sizes in this sampling. So they had some reason to think this might be reasonable. And I think it is a possible thing in this narrow phylogenetic region. If we look in the broad data set, that correlation does not hold true at all. So this is sort of mean cell solidity versus the mean cell area, just sort of log transform for niceness. And there's no real correlation here in our large data set. So what that might imply is that, if you go back to this tree where, potentially we have some signature, let's say sort of here there's a little bit of signature for some type of undulation, that might have been optimized to say minimize area and stress, geometric stress. But over here where you have some correlation, that could have been optimized for a completely different function. And so the idea is, depending on where you're growing, how you grow, all of the other constraints on your growth and development, you may or may not have chosen to optimize whether or not you have loby cells as one of your calculations for fitness. And it may have been useful in different plants for different reasons. So in some, it might be useful for mechanical integrity because they grow in really windy environments where the leaves have to flex a lot. And in others, maybe the geometric stability of the shape was more important during the evolution of that part of the plant. So it's what we think based on this, but again, we need to do more in-depth analysis. It seems as though there's probably not one single function that was optimized, probably not a single function that got optimized multiple times, but perhaps multiple functions in multiple locations. Okay, I don't really know how much time I have left. I'm going to point out that our measures really aren't good enough yet. So solidity is not the best way, really, to describe margin undulation. There's problems with it. As an example, this is that lowest quartile of data, the sort of 25 gram median, and you can see, okay, I understand why this would be sort of called undulate, but I think, why is this guy here? Or this one? Well, obviously, the mathematicians will know that this will give you a solidity value that is low because of this single curve, this gap. So we wanted to find better ways, and you may also have suggestions of ways to measure this, but we've started looking at total absolute curvature along the path. So this was at an NSF workshop. I was working with a PhD student who's at MIT, and he said, oh, well, I've done this for rivers, maybe we could do the same thing for cell shapes. So it's more like taking examples from geography. There's really nice things about this type of measure, which I probably don't have to convince you about, but one of the things I think could also be really interesting is that we might be able to carry this onto surfaces instead of just two-dimensional shapes. So here's an example. This is, I call it capybara, because it's capybara, and I like capybaras. So essentially what we do is we take our individual cells. We're going to fit, there's an optimization function for how many points at which you need to calculate curvature along the line. And you can imagine that the number of points would also be a proxy for how undulatory your margin might be. So you can use that as a metric. We then take roughly 30 samples per species and sum that, so you get kappa bar bar, which is capybaras and bar. Okay. The distribution is, again, very long-tailed, very highly skewed towards less curved things, so more smooth margins. And so we're developing this a little bit further, and we want to see if we put this onto the phylogeny.
So this is across species?
This is all the species, yeah, that we ran in. So this is the distribution of capybara bar, so the summed total accumulated curvature within species, but across all of the species. Yeah. So again, most things have, this is sort of number of species, for example, most things have very low undulation, high undulations by this metric. Okay, so I'm going to tell you, yes.
If I were to look at traits that people suspect have some fitness effect across these different species and plants, what would the distribution of them look like? Would they mostly look like undulation, or would they mostly look more normal-ish, or would they mostly look?
I have no idea. So the question is, if you were to look at traits that people think correlate to fitness in plants, would they tend to have a distribution that was sort of long-tailed like this, or would they a one-sided long-tail, or would they be normally distributed?
Well, and a lot of traits are under sort of stabilizing selection, which would suggest
should
more
give them a
normal. But yeah.
I mean, the other thing is that I think it's highly probable, so I'm going to get to some ideas of mechanism in a second, but a lot of this is underlaid by, say, the actin cytoskeleton. And you can imagine that that could be under selection for a lot of reasons, and that what you're looking at here is just a random thing that happens to happen if you change it for some other purpose. And so this trait changing over time may be because it's actually rarely under selection. But then, yeah, I don't know if that's helpful at all. It's interesting to hear from you people as well, not just the mathematicians, but also people who think about evolution, because this is sort of different. And what questions are interesting to look at. Okay, so if we just step back for a second and say, okay, well, when you have undulation, how do you get it and what does it look like? So this is sort of this idea of morphogenesis of shape. And we have some ideas of how this gets generated in model species. So I'm going to most specifically talk about maize. You start off with a square, that square starts to elongate, and I'll show you some data for this in a second. And then it elongates even more, and at some point the margins start undulating as well. And please note that these undulations are plastic. It's not an elastic undulation. So if you sort of took out the cell contents, those walls would stay like that. They're getting fixed. So the, sort of what we're interested in is how this happens, and there's quite a bit of evidence that, this question we look at separately, but here, basically what you're doing is you're depositing more soft material at one point, and you're sort of putting some more rigid material at this point, which gives you constriction versus an expansion. And there's one model that exists right now in the world that says, you have protein A here and protein B here that help these two processes, and if you model them as reaction diffusion, you can get a pattern. So you get a pattern of undulation based on mutual exclusion.
If, as I thought you said, it's moving the third, this is also changing the
Yes.
third dimension, do you know whether, even though it's pinched overall at those locations, whether it's, when it's convex versus concave, is the pinch larger or smaller?
So it's a question about how the 2D undulation geometry relates to the 3-dimensional geometry on the pinching. And we do not know that yet, because everything that's been done so far has been on 2-dimensional.
No, but your images surely show that to me.
Yes. So, but we haven't quantified it. So we have confocal data, for example, for maize, where we can actually reconstruct a mesh around a 3-dimensional cell volume and look at surface curvature. We just haven't done it yet. I don't have anyone that wants to work on this.
But even qualitatively, I think if you just look at the
Yeah.
images, you should be able to immediately tell whether the convex region is thicker or thinner.
Do you want to,
No, no,
you can
later.
look at it now. Here you go.
Eyeball it.
Okay, so, yeah.
And you think would be helpful to understand how your kappa bar evolves in time and constrain that with the stretching, so how much stretching
Very
you have on the
much.
contour?
Very much so. So the question is, if you want to, so if you think kappa bar is sort of a good measure, we might like to look at how that evolves during morphogenesis, and then what constraints might exist on that number, and how it correlates to other things like growth rate of the wall,
Yeah.
elasticity of the wall. That is exactly what I would like to do. Yeah, I just need to find it if I can't do that. Okay.
Wouldn't you want A to be soft too, so the neighboring cell can push in and make
Oh, yeah.
its own?
That's like, so the question is, wouldn't A need to be soft as well, because the cell on the other side is sort of pushing? And I think that's a major flaw in sort of one of the cell biology models, which is that they say, well, this isn't growing because I'm restraining it somehow, but you're exactly right. So this is growing, but this is also growing. It's just growing in a different, like,
Can you watch it grow and show that both sides are moving, or both curves are moving?
that would be wonderful. We have not done that yet, but we should be able to. One of the things that would be really good to do were you to do that is also have some sort of like fiduciary marks that you could follow. It's kind of, there's one example I can tell you about in Arabidopsis. In maize, one way people might do that is scatter fluorescent particles on the surface, where you could imagine if you have a fluorescent dye that you could bleach areas and use those as fiduciary markers, but it could be more damaging. So people have done this in Arabidopsis, which is this other model species that has puzzle piece shaped cells, but it is, I think it's fundamentally a less tractable system, simply because the shapes show less regularity. If you look at the number of lobes and the degree of lobiness and things like this in Arabidopsis, it's almost a completely random thing that happens. Whereas you have a little bit more uniformity in the maize, probably because of the shape before interpreting undulation. Okay, it could just tell me what to say. So this is a snapshot of kind of what the cell shapes look like as they're moving through time. And in maize you can do this nicely because a single leaf actually is a developmental time series. New cells are sort of formed at the base of the leaf, and the older cells are near the tip of the leaf here. So you get this nice developmental time series. Yeah.
why aren't there stomatas here? Like, how do they take in or like exchange
Yeah.
when they're growing?
So there's stomatas here, for example, but not here and not here. So actually, so these cells, this is going to become a stomata. This will probably become a stomata. And then so the stomatal pair gets born in developmental time as well. They're not all here in the beginning.
So they can still exchange air because the walls are different, or?
Well, if you think about it, this leaf doesn't live, those cells don't live in isolation.
I see.
So they're probably not very photosynthetically active. In a maize plant, they're really wrapped up actually way down inside, so they're not photosynthesizing yet anyways. But in terms of sort of how you grow shapes when they're actually physically stuck together, that adds in another level of complexity, that you have this other cell type that's getting born and it's doing things, and how do you accommodate that as well. So I hope, you can kind of see here that they are relatively cubish, sort of down at the base, and then they start elongating, and then they start undulating. That's sort of the gist here. This is as they're getting even older towards the tip, and you start seeing this beautiful undulation. Okay. So what's interesting is there's examples where the undulation is different. So in maize, and in all plants actually, you have what are called juvenile leaves. Some of the first that are formed have some characteristics that are different than adult leaves. This is like puberty for the plant, although it has nothing to do with sexual maturation. It's just that they switch some of their morphological characters. And so in a juvenile leaf, you have sort of a weak undulation, even though this is about 100 microns. The cell length isn't actually that different, the final cell length near the tip of the leaf, but the degree of undulation is very different. So that means it's not scaling. It's not scaling.
Is there some kind of maximum adult undulation that is achieved, and then no matter how old they get, they won't change anymore?
I would say yes, except we actually haven't done our new sort of capybara type metrics on the maize data set yet. So now I'm going to show you some very qualitative stuff we want to apply the quantitative analysis to. What's interesting is here, this picture doesn't show it, but I think a little bit older, you actually start to see undulations upon undulations, almost in a fractal type way, which I think is interesting. So I don't have a picture for that here in this presentation, but it's sort of, a low level, it's the frequency is, you're getting more undulation and then undulations upon undulations. Yeah. Stop. Okay. So.
Just had one question. Do cells divide or just grow?
This is a good question. So it depends. So in the maize leaf, there's division in the beginning, but it stops quite quickly, and then it's mostly elongation. So when a plant cell wants to grow, the cheapest way to grow actually is to just pull in water and expand your volume and distend your wall. Whereas the synthesis of a new wall and synthesis of cytoplasm is more expensive. Most things that we think need to go quickly and grow very anisotropically tend to do it through this type of anisotropic elongation. But there are examples where we have division, and we might be interested in how that relates to undulation as well. There's also variation within a single leaf. So this leaf is the same age. We're looking at cells that were born at the same time, but there's less undulation here, more undulation here. There's also different cell shapes that you can see.
And sorry, this where is the mid part of the leaf?
So this is very much, so we're up near the tip, and this is probably kind of a little area right here. In maize, you have parallel venation, so you have a lot of veins running up here, and you're tending to have,
so more.
There's going to be a vein running underneath here, so there's possibly correlation between tissue levels.
And more undulatory near the center of the leaf and less towards the periphery?
No. So the pattern does not appear to be, at least in maize, there's no sort of global patterning. It tends to be more locally related to the anatomy.
Sorry, in this undulation direction perpendicular to growth direction?
Is the undulation direction perpendicular to growth direction? So these cells are growing, so the outer face and the side face and the bottom face are all growing. The end plates are not. And they're growing specifically more in one end direction than another. And the undulation is perpendicular to that.
how about for other plants?
So there are lots of examples of plants that grow like this but don't undulate, and I'm trying to think. I don't think I can answer that question directly at this time. So things that look like they're growing more isometrically tend to have more, they might have more non-directional undulation, but I think the growth is important to develop the undulations. But whether the
So if you were to project stress,
yes.
right, as the leaf gets bigger, as it grows, or subject to wind, as you mentioned, and other things, even locally, do you see there is some kind of correlation between? I'm specifically thinking about, we study sutures in the skulls, and they're there to dissipate stress. And they kind of, in morphology, there's some of these things is
Looks
the
like a
micro
suture,
similar,
right?
exactly. So are they there at least partially for the same, to do the same function?
It is possible that in some cases that is a mechanical stability optimization. There are some experiments. We have a mutant in maize that does not undulate. It's an actin mutant, so it probably has a lot of other problems. But we could test, for example, mechanical integrity. But it gets complicated because other things are changing in the background. So I like the mechanical integrity idea.
Although that's more at the supracellular level, right? In other words, the sutures are not, are the undulations at the cell level?
So they're supras, yes, but they're kind of microscopic for sure. But again, you can imagine the force could be at different scales as well. There could be similar tensile or compression forces. At the cell level,
There's a theory in plants where you tend to, they believe you have more tension in your epidermis than you do in your cortical internal cell layers, so that the epidermis sort of constrains growth, and so it's constantly sort of under tension. And so if you had a growing system that was under tension, but then also, giving torsion and all sorts of weird things from the environment, it may be advantageous to have a more stable interface. But we haven't been able to test it. We should be able to, it's just a little complicated. So I'm going to leave you with this because you don't get to hear about circumvention. You never get to hear about that. That this works, but I don't know. So this is our confocal stack, and we're moving from the bottom up. And it's going to loop, so you can sort of see what my point is in a moment. We're at the top surface of the cells, the free surface, and they're undulating. But if we loop back and we're coming from the bottom again, so those are the cells in the internal layers. And hopefully we're going to start coming through our cells again, and we don't have to get the bag. So I hope that you notice that the lower margins are straight, and also that the cells appear to be getting wider up towards the top, which essentially means that the pattern is more beautifully complex in terms of its generation as well. So, just for fun's sake, a really crappy elongated rectangle. Essentially what we're saying is that your undulations are happening up here on these walls. They're not happening on these walls. They're also not happening down here. And there is an increase in the width dimension up here, so width upper is larger than width lower. And so we can
And
ask
furthermore, it's like this.
Yeah.
It's
So we can ask some interesting questions about how does that three-dimensional shape change over time? Whether we can look at differential mechanical properties that could lead to this type of shape, and also this goes to this three-dimensional idea. It's possible that these margins undulate because this wall is doing something that causes them to pull. And so I think there's some interesting questions there that sort of deal with the physical nature of generating these undulations.
And this is also what you see in Arabidopsis?
No. So in Arabidopsis, the bottom and top match. So there are examples.
There's not even a phase?
No,
No?
they're completely matched. Yeah. And so
are they straight leaves or like curved leaves? Like if you have an overall curvature to the leaves, then it makes sense for the shapes or the properties of the.
maybe if the overall curvature of the leaf was actually felt at the scale of a single cell, right? So you would need to be able to feel the global curvature at the local level. It's possible, but I expect that you never have that extreme global curvature that would make that locally.
Curvature is local.
Sorry, yes. I guess I'm on an organ level versus the cell level. Thank you. Hi.
Could you sort of test these ideas of what the forces are actually doing? Like, even if you just had the leaf grow across like a flat plate, or if you pulled on it while it happens, you could start.
Can you test these ideas about what you think physical forces do?
This is your last slide.
Yeah, I'm gonna stop. So, yeah,
These are the questions.
these are the questions. We can talk
And this
about
will be
that.
the last one.
You should, in theory, and someone has done this in another species for a completely different reason. Like, just take a wire and glue it as the leaf is growing, and allow it to apply some tensional stress and then potentially also compressional stress in the other direction, and see how that influences the evolution of these shapes. I would love to do that. So we just moved our lab to Los Angeles, and so in the movie we might have been a bit disrupted. But we want to test these ideas, and we want to look in, a genetically related group of plants that has ecological and form diversity, and ask whether they also show some shape diversity. So,
I want to just thank hopefully we can reconvene at 2:00.